What if the answer to whether we can create new life depends on how we define life itself? Despite decades of study, biologists still lack a single universal definition. Even at A-level, life is taught through a biochemical lens that reflects only one mechanistic viewpoint. Across scientific fields, the definitions shift, exposing a deeper issue. No consensus exists. For this project, we use NASA's operational definition because it offers a clear, practical framework. But the definition you choose shapes the conclusion you reach. In other words, the question, can new life be created, is inseparable from the question, what is life? Also, what counts as new life is largely a matter of perspective. Biologically, newness isn't inherent to an organism, but reflects human discovery. Discovery is not equivalent to creation, as species can be newly observed, yet biologically ancient. Furthermore, reproduction isn't considered the creation of new life, but simply the continuation of existing life. Offspring inherit the same fundamental genome structure through recombination of parental DNA, producing unique individuals within an existing species. Even mutations are just alterations of an existing genome, creating variation but not a truly novel life form. Deviating away from reproduction, some experiments have tried to create new life on the basis of theories that propose life can emerge spontaneously under suitable conditions. Early models assumed these conditions were a mixture of various elements, like a primordial soup, allowing components of living systems to form gradually. The Miller-Urey experiment modeled early Earth by stimulating these elements with electric sparks. The results were promising. Amino acids, urea, and hydroxy acids were formed spontaneously. However, these molecules were neither self-sustaining nor capable of Darwinian evolution, and thus do not meet the definition of life. They represent only prebiotic chemistry. This led us to think, if all life share a single origin, is synthetic creation the only way to genuinely create new life?
For example, if we assemble known components such as DNA sequences or organelles, are we genuinely creators or simply replicators? Craig Venter's artificial bacterium experiment illustrates this dilemma. Although its genome was synthetically assembled, it relied entirely on pre-existing biological systems. But creation is not just assembly. It requires designing and initiating life in a way that produces autonomous, self-sustaining systems. Advances in digital life and AI have produced systems unlike any biological organism. Self-replicating and mutating programs, such as those in the 1991 Tierra platform, can evolve, compete, and generate new behaviors. Unlike Venter's synthetic bacteria, their variation arises entirely within a computational substrate. But despite mimicking evolution, these systems are not self-sustaining. They depend on human-provided energy, cooling, and maintenance, processes that living organisms handle autonomously through homeostasis. Digital systems therefore simulate aspects of life, but lack the biochemical independence required to be considered alive. So our current technology can assemble, mimic, and experiment, but it cannot yet create a truly new living system. However, even if we can create life, should we? Constructing life is not a neutral act. It carries unpredictable consequences, from ecological disruption to biosecurity risks and unexplained evolutionary paths, emphasizing the magnitude and ambiguity of these interventions. The idea of playing God further heightens the ethical debate. Does creating life cross a moral boundary? On top of that, the life we create does not consent to exist. If it suffers or fails, do we have the right to terminate it, or must it remain confined to a laboratory? These questions underscore the ethical responsibility tied to any attempt to create life. Ultimately, creating life does not simply add something new to the world. It fundamentally alters biodiversity. Once life becomes something that can be engineered rather than discovered, its meaning threatens to shift from a phenomenon to a resource we design and potentially discard. With current technological advances, the ability to manufacture new artificial organisms may soon be possible. But without a universal definition of life, it becomes increasingly difficult to determine what even counts and identify what we've made. In this sense, creating life may not be a biological milestone, but a conceptual one, where the act of redefining life becomes part of creating it. We are stuck in a loop. We can't define life without the created entity, and we can't identify the entity without the definition of life. Only when we break free from the circular debate can we stop asking, is this alive? and instead determine what we intend to build, finally giving a definite answer to the question of whether new life can and should be created.